Mike, I'll just introduce Sara Wells. So Sara is director of the Mary Lyons Centre in Harwell, and also plays a leading role in the National Mouse Genetics Network. So this is another network that's really runs in a quite parallel way to ours, but is obviously exploring gene function in the context of another type of model system. So over to you, Sara.
Thank you very much. I've got new glasses, and I have not quite worked out the focal length between that, and so if I step back some falls and put my glasses up and down, please excuse me. Thanks ever so much, Jonathan, for inviting us here. I'm representing the National Mouse Genetics Network, and we were one of the first types of these networks that the MRC launched in an attempt to get people together who were sort of the best in their fields, but dispersed into different organisations together into what is a functional network of people working together and exploring things that you might not have got chance to explore if you hadn't been put together in one of these networks. And for those of you, it's been really interesting working with Jonathan since the start of the Functional Genomics Network, because there are a lot of parallels. We run slightly differently, but there's lots of functional genomics work going on within the Mouse Genetics Network. And hopefully for you guys, when you look to model organisms, some of them might be a mammalian organism such as a mouse, and you'll look to us to support that. So I just want to tell you a little bit about how it's set up and the work we've been doing and some of the things we've done so far. So we had a £22 million investment at the start. That's for 5 years, by the way, not every year, which would be a dream, and we could spend it, but it's not. But that's not the case. So 22 million at the beginning of the network, launched in April 2022. We'd obviously done a bit of work beforehand to get ready for that. Unfortunately, probably not enough on our contracts. So, but we launched in April 2022, and we had an additional ageing cluster. In some miracle of logic that we don't quite understand, the ageing cluster came last, but a cluster working on ageing animals in February 2024. And these were the clusters that were selected and put together were really to capitalise on the UK's strengths and in developing new complex models of disease. So we've also got lots of strengths in single genetics and in single genes and in less complex, but these are complex genes and complex phenotyping. And one of the things that's always been at the centre of this is an integration of well-annotated human data, but working alongside human cohorts and human data sets to ensure that the models that we're developing are reflective of what we're seeing in human clinics. We are capitalising on advances in disease phenotyping, so we're developing lots of different types of phenotyping, especially mouse phenotyping, looking at its relevance to what happens in human clinics and whether we can actually model the same biomarkers that we're modelling in human clinics and whether they mean the same. We're very conscious we're on a different species. The hub of this is the Mary Lyons Centre, which I'm the director of, which is on the Harwell campus, just south of Oxford. And our key challenge was to set up predictive models of disease. So these are our 8 clusters that, as you can see, are scattered around the country with multiple cluster members in multiple different institutes. And you might say, well, these are loads of people all over, but remarkably, we get on remarkably well. We have big meetings together, we have fun meetings together, and there's a lot of crosstalk in between the meetings we have together. There is an ageing cluster, a cancer cluster. I'm not going to read all of these out, apart from a congenital abnormalities cluster. And if you go to poster 2, Karen and Steve are there to show you what activities they've been doing on congenital abnormalities. There is a technology cluster, which is working around DEGROM. There is blood cancer clusters, the microbiome, which has given us, I'll talk a little bit about it, which has given us a kind of initiative to be able to expand the technical repertoire of the Mary Lyons Centre, mitochondrial, and a cluster looking at juvenile behaviour and early life behaviour. These are just all of our photos. I just show this because it's a nice photo of me where I don't look like my mum when I brush my hair. So, but these are all, and the 2 on the bottom right are here today. And our project manager, Lyons, is here. Where are you, Lyons? He's here, right at the back there, and he's got a table out there, so he can tell you all about accessing us and working with the Mouse Network. The Mary Lyons Centre, as I've said, is at the centre of this, is the hub at the network, and we are the MRC's national facility for mouse genetics. So our whole remit for being there is to enable research. We have technical development research ourselves, but we have no disease-disciplined research. We're completely driven by the scientific community. And we help with in vivo aspects of research programmes, both in academia and bio and pharma. And we support a lot of people nowadays, we support a lot of people where mouse genetics isn't the core part of their program, but it's a part of their program that they need to integrate for certain questions. So these are people that are non-mouse experts that want to access the resources that we have. We have a training centre, and we use the training centre for a lot of our meetings. You're welcome to use it too. And we use this to disseminate the best practice. And actually within the network, we've had lots of workshops, and we've got several planned workshops on the specialist techniques within the clusters to make sure that we're teaching people outside of the clusters things that we're developing. It's also really important that if you are a national facility that you contribute to setting standards, and setting standards for quality control of new GA animals, for example. And some of you might have seen there's some new publication guidelines that we led on that tell you what you should publish when you're publishing details about new mouse strains that you're generating. Just to say that these are all of our clusters, and on here you'll notice it's a non-funded cluster, but it's an associate cluster, which is the UK DRI, and they sort of join in and do some work with us as well. But these all centre around some common interests in deep phenotyping and technological advances in the data, and Crispin Miller's here, who heads our data portal up. We have big training hubs, and we're really focused on great model development, but a great integration of information from human cohorts and human studies into what we can do in terms of mouse modelling, and then using that data to match back onto patients. So we've been going for 3 years now, so I thought I'd show you a few of our kind of key performance indicators. We've got over close to 500 associate members, and these are people who follow our newsletters and get in touch with us, and who we can ask for specialist things outside of the remit, specialist advice outside of the remit of the network itself. And we have 14 industrial projects currently running. We've had upwards of 50 events where we've organised events like this. This would be one of our bigger ones, but events from smaller events to ECR targeted events, to events where we're teaching technical procedures and details. We have over 120 in our ECR network. We've recently launched some webinars, and we have a PhD boot camp where people who are new to their PhD or new to their postdoc, maybe not having used mouse research before, come and spend a whole week with us at Harwell, and we do work you quite hard. That's why we call it boot camp. We've generated or in the generating over 50 new mouse models, and we've gathered some additional funding from additional partners and granting, and we have lots of technology transfer visits where people from the hub go out to each of the cluster labs, or the cluster labs swap over people to make sure that we're maintaining this big training network. So our kind of highlights have been getting the extra cluster. It's always nice to get an extra cluster, and because aging sits across so many of our clusters as well, this was a really good opportunity for us to bring together some of that expertise into a bigger cluster with a more focused speciality. We had a business engagement from the MRC, where the MRC gave us some money, and we looked for partners where there were matched partnerships in industry. So the MRC contributed 700,000, and we'll get about 750,000 in kind contributions from industry to run some very focused projects on our models or on our speciality. We had a capital bid, which is going to allow us to do some imaging. And this is at the Cancer Research Institute in the Scottish Institute, and we had a data platform which we had advisory groups that helped us to refine the planning for, and we're now ingressing data from the clusters to a kind of central point. It's quite difficult data, the mouse network data. I'm sure all data is quite difficult, but it really ranges from sequence data right the way down to video data of mice and everything in between, including every assay you can imagine under the sun. And some of it is only 1 or 2 assays that will be done on certain models, which means you've still got to do the same kind of bioinformatics infrastructure for those 1 or 2 assays. So this is quite a challenge for us. I think the big highlights have been, especially for the Mary Lyons Centre as a hub, have been the fact that we've established these project teams. We have very regular meetings with the clusters. We have effective communications. We work very hard at effective communications to make sure that all levels are communicating and communicating well. And the Central Hub participates in the working groups, and we have a number of working groups that look at things like like best model systems. So not only did we work on the mouse, but we looked to reach out to other model systems, such as flies and worms and organoids and in silico modelling, so that we make sure that we're actually picking the best model for the question that we have in mind. We've had a huge progress in our experimental platforms. So from the hub and also from the clusters themselves, we've been able to expand our technologies and expand the number of techniques that we can perform and we can do. We've also collected in lots of different colonies of mice that are now from all of the different cluster members that are now open to the community and that go through the public archive, which is hosted by Harwell. We have lots and lots of technical skills exchange. I can't tell you how valuable being part of this network has been in us being able to learn and us being able to teach different techniques, right the way from molecular techniques to imaging and animal care. And we've had a lot of discussions over animal care with the different facilities that we're sending mice to or that we're working with. And the data platform, we have a data coordination centre now established in ingressing data. So that's been quite a big highlight. And we have, well, actually they're finished now, the mouse metadata parameters. You wouldn't believe how many ways there is of describing a mouse and where they come from. It's taken months. So I guess, you know, we're kind of a couple of years ahead of the functional genomics network, so I thought it would be remiss if I said everything's been dandy all of the way. Some of the things that have been slightly more challenging is that it is a different approach, and it is a different approach for a central facility, and the project management has been, I wouldn't say it's been a challenge because actually we've really enjoyed it, but we've had to employ certain different types of staff, and staff that are outward facing and that are credible and that can bring people together, rather than some staff that we might have employed in the future that would be very technical focused. So it's kind of a different way of managing things. I don't want to go into IP and MTAs, but to say that they are complete and utter bore, and that the more that our technologies move on, the more we are absolutely stifled as a scientific community, especially when we try to work along pharma and biotech with all of our technology licenses and every university in the world thinking that their mouse is going to make them £1 million. It's really not, and we can make it ourselves if we wanted to. We just want you to give you yours. So, you know, I think as a community we need to think about how we handle IP and MTAs easier, because it debilitates us and we spend too much resources on it. The data platform came on later, and you're lucky that you have yours up front. And I think when we apply for a refund, it's an integral part of all of these networks. If you're collecting data at 100 places, you've got to have somewhere that can bring those 100 sets of data together, and that's really important. And I think the technical training has been a steep uplift from moving into different models, especially for a service team. We've really enjoyed it, but it's been a steep learning curve. So this is what we're developing at the moment, and this is how you guys will be able to access our mouse models and our protocols, and eventually our data portal, which will be available for people to take unified data sets that hopefully they can then compare to the data sets they're getting out of your functional genomics. And that will be really interesting. And those unified data sets will have all of the information about the mouse, about the genotypes of the mice, as well as what's done with them. And there will be an easy format, hopefully, that you can do some clever bioinformatics and see where our modelling maps and where it doesn't map, which is just as important. So in our future plans, and this just said, we're obviously going to carry on developing our new in vivo and alongside that in vitro systems for modelling human data sets. And we're using data sets to inform these models. So in the past, we've kind of made a mouse model and then seen where it maps to a disease. It's very much the other way around now. And we try to model, and lots of our models also are oligogenic, so we have lots of different gene perturbations and protein perturbations going on in the same animal. We want to align these, obviously, to the human data sources. We're working much more across clusters, so bringing these kind of clusters that didn't necessarily have a massively common theme apart from the mouse together has meant that we're able to work across clusters for some really complex questions that you normally wouldn't or would be quite difficult to tackle outside of one of these. And things like, you know, the effects of mitochondria on blood cancer, early life influences to adult diseases, and ageing, and how does that affect all of our clusters and all of these wide-ranging effects. It's really important for the Mary Lyons Centre to deliver not just to the cluster, but we develop techniques with the clusters that are then available to a much wider academic and pharma community. So this has been really important for us. And some examples of this are, we have a germ-free facility set up with help by the microbiome cluster at Harwell, so at the Mary Lyons Centre, so you can get access to germ-free mice if anybody's doing microbiome studies. There's also a spatial metabolomics set up at the Scottish Institute, which is accessible. So this has really been, what we're really heading towards is a real step change in the way that mice are phenotyped to increasing relevance. And we're now looking at things like change in temperature, change in the way we feed mice, trying to mirror much more the human condition. And this is really where we're going in the future. And I thought I would just finish because typically people go, oh, mouse geneticists only ever work on one background, and they're not in the context of a diverse human background, especially this community. And just to say that investigating a much wider range of genetic backgrounds is already happening and is being part of our future plans to look at what modulation different backgrounds and different background strains have on the phenotypes that we're seeing. So just to say thank you to everybody here, and this is all the cluster leads and everybody meeting up, but most of all, thank you to these fellas. Thank you.
Thank you, Sarah. And I must just say personal word of thanks because, you know, we're learning from the things that you've gone through as a network. It's been really invaluable for us and very much looking forward to sort of partnering between the initiatives.